So an enzyme is a biological catalyst, and I like to imagine it like your hands are the enzymes, and what it does is it arranges the molecules in unusual ways in order for it to come apart or to join together. And so that's the induced fit model. So it is increasing the reaction rate because the particles collide has just to do with concentration, which is not what we're doing. We're doing temperature, but they will collide more often if you increase the temperature. So the activation energy is helped by temperature because increasing the temperature can give the molecules more energy to collide and break apart. But the enzyme also helps the reaction occur faster because the activation energy is lowered by the enzyme. And so it can react faster, and it lowers the activation energy by the induced fit metal. It grabs onto it. The positive and negative areas of the that little shape there matches the substrate, and so it attracts the substrate particles to it and sucks them in. But it doesn't fit exactly right, and so when it sucks it in, it pulls it apart slightly in the areas where it is asked to be broken. Which is its job to break it into certain places. And so that's the enzyme lowering the activation energy. The temperature, increasing the temperature also gives the molecules more energy in order to break itself as well. And the enzyme also helps as well as temperature as well. So the temperature increases the collision number of collisions, so the probability of colliding in the correct 3D orientation increases. But the enzyme also holds it in the correct place. That's what a catalyst does, so it breaks. And so there's quite a lot going on in this. Its temperature does affect 1, 2, and 3, but the enzyme has a massive impact on 2 and 3, sometimes like 1,000,000 times greater. So this is how the reaction goes. I just mix I think it's maybe 5 grams in 500 mils, and then I make up a sugar concentration so that when I add 10 mils of both, it'll be about 15%. And so I make sure that's dissolved. And so I incubate both. So I get annoyed at students because they'd give me readings above 50 when the enzymes don't work above 50. And so I've got all the water baths there. I've got 10 grams of the sucrose ready to incubate. So I'm 10 grams into 3 of the tubes. So I'm only doing a repetition of 3 because it just takes too long. Eventually I give up because the whole thing just takes too long. And then putting 10 mils of yeast stock solution as well. And so the same stock solution of sucrose and yeast in all the 6 water baths. This one's sitting at 10 degrees, and so I'm just waiting for those to equal 10. So with my last one, I put a thermometer inside the actual solution to make sure it reaches it. And so then I put the denser liquid glucose over the top so it sinks and mixes, and then give it a bit of a spin. So you flick it to spin. Then I put a balloon on it and just wait. And that's it there. It takes too long. I think I waited 1 or 2 hours. I got impatient after half an hour and took readings at half an hour, and that's what we're going to go with. It's on an angle, I know. It's not very good. Could have gone stars, 1, 2, 3 plus stars. And so I just got the ruler out. Uncertainty before and after, so that's 1 mil. So 1 plus or minus 2 millimeters, plus or minus 1 millimeter for both. And you can see, even though it does work quite well, it's not good enough to fill up the balloons in an hour or 2, so not good enough. And so that's the data I got. It is better it's kind of the reverse of what I was hoping. So the temperature graph is on the left here, and it should slowly increase due to rates of reaction and then quickly drop off because the temperature, the kinetic energy disrupts the bonds and destroys the enzyme. It gets denatured. Similar to the pH graph on the right. So both sides drop off pretty quick because of getting denatured on both sides. And so I got a graph, but not the sort of one I wanted and not good enough. And so improvements for that, of course, if you're doing an experiment seriously, you should always use a data logger. So the carbon dioxide data loggers would have worked perfectly, and probably within 30 seconds even. Well, that's not true. It might take 5 minutes for it to activate, but certainly within half an hour would have got amazing results. So if you're doing experiments properly, use data loggers time was an issue for me. Increasing the volumes is going to increase my time and waste materials, so I'm not going for that. So with that picture out, but increments of 5 degrees Celsius would have also helped get a really accurate picture. Okay, so this is why I don't do it very often. We just don't have time, guys. That took quite a few hours, even though this video is quite short. Okay.